All right, I'm live. So we're going to review Bio 303, Test 4, or the second test in the evolution class. And this is going to cover a lot of phylogenetics, and it's going to cover Earth history, starting from the origins of Earth all the way to modern diversity. And what we can also cover are the great inventions or the great evolutionary innovations leading to modern day diversity, and also cover some diversity as well that we have today. So I'm going to go shut my door, and anybody's got a question, I believe you can start typing in a question now, and I will answer it for you. Okay. Live chat. Anybody got a question here? I know you guys have some. Here we go. What are the recent observations that support metabolism first theory over replication first? And that's a really good question. And this goes back to the theories of abiogenesis, and we covered three different theories. There's actually a fourth one that was disproved over several hundred years ago, and that was spontaneous generation. But other than that, the modern theories are the Miller-Urey experiments, which is a prebiotic soup model, the RNA world hypothesis, which is the self-replicating molecule, and then the third one would be the metabolism first theory. So the recent observations that support the metabolism first theory are this. Think about this. Every single living organism on this planet, has some things in common. One of them is life is a system out of equilibrium with its environment. Life is an island of low entropy. We are ordered, and every living organism extracts energy from its environment to create order and maintain homeostasis. So, if life is a system out of equilibrium, well, where in nature might we find systems naturally out of equilibrium? Now, let's take some things that all life has in common besides just this general thing about what does life do. The next thing is, every single organism on this planet makes ATP using chemiosmosis. And to make ATP using chemiosmosis, of course, this goes with by creating a proton gradient. So now we go, okay, every single living organism on this planet is creating a proton gradient across its membrane. So somewhere, if we say every living organism does this, then we can infer that our common ancestor also made ATP, which is the energy currency of life, by chemiosmosis using a proton gradient. So these observations lead us to believe that metabolism, the sum of chemical reactions that create organic molecules and concentrate them, probably came first before self-replicating molecules. Because the problem with the RNA world hypothesis, there's several problems with it. First of all, you go from a self-replicating molecule to a cell. How does that happen? How do you get a system out of equilibrium? And secondly, if you don't concentrate your RNA molecules, if you don't concentrate them, then they're only going to get up to about 50 nucleotides long, and that's it. They're not going to get any bigger than that. So there comes this limit to how complex these molecules are going to get. Okay, hope that answers that one. You guys are really hitting it fast now, so let me go through the next one. Why is life an extension of geological processes? That's exactly right. If you think about it this way, all right, you've got these geological processes. You've got a water cycle. You've got a carbon cycle. You've got a phosphorus cycle. We have all these cycles. And let's just follow carbon, for instance. Carbon is an element, and it goes back and forth between a biotic and abiotic pool, meaning there's carbon inside your body. That's organic carbon. It's in the form of carbohydrates and lipids and things like that, and other macromolecules. Well, every carbon atom in your body was fixed by a plant, and it was originally CO2. And that CO2 may have been expired by another living organism. That CO2 may have been released from a volcano. It may have been released from rock weathering. So carbon, for instance, is going to go back and forth between like rocks, coal and oil, into carbon dioxide in the atmosphere. It's going to get fixed into organic molecules. From there, it can be switched back into carbon dioxide again, or get put back into rocks. So that's one of the reasons why life is an extension of geological processes, because every element in your body is going to get recycled. And it's going to get recycled and go back and forth between organic or living reservoirs, which is life, and inorganic reservoirs, which is, gases in the atmosphere or rocks in the ground. Okay. Kayla, can you explain why oxygen is important for Earth more than just breathing? Oh, yeah, that's a really good one. Okay. We get this idea that all life requires oxygen. Well, of course, we require elemental oxygen because, every macromolecule basically, or except for some lipids on it, has oxygen attached to it. But when we talk about oxygen, most of us are thinking of O2, which is molecular oxygen in the atmosphere. And of course, that's required for basically every single animal to breathe. Not all life, because not all bacteria life requires oxygen. But oxygen is very important for multiple reasons. One of those reasons is that it cleared out the atmosphere and removed a lot of the methane and other organic molecules in the atmosphere. It also rusted out our oceans, and we achieved the clear oceans we have today. But importantly, when oxygen got in the atmosphere, it formed the ozone layer, which protects the surface of the Earth from ultraviolet light. That is important. If we didn't have an ozone layer, then no living organism was going to really survive on the surface of the planet. I'm talking out of the water, because water can filter filter ultraviolet light. So it would prevent life from going onto the surface. The other problem without having oxygen in the atmosphere is that the ultraviolet light can actually split water. And the Earth is not quite large enough to hold on to its hydrogen gas. So eventually, over billions of years, Earth would have lost its water and become a dry, barren, oxidized planet like Mars. So that's why one of the reasons why oxygen is so important, because it helped to, stabilize the water on our planet by forming the ozone layer. And of course, the other very important thing about oxygen is that, of course, it energized life. Oxygen is the second most electronegative element in the universe. And because of that, it's a great electron acceptor. And what that means is if every living organism is making ATP through chemiosmosis, then by using oxygen as your final electron acceptor from your electron transport chain, that means you can get a lot more energy out of the organic molecules that you started with. So cellular respiration became incredibly more efficient, providing a lot more energy to life. So in a way, we can simply think of this as oxygen energized life on this planet, and it paved the way for the origins of eukaryotic cells, and it paved the way for multicellular organisms, including plants, animals, and fungus, and made our lifestyle, our very active animal lifestyle, possible. Okay. How is the origin of eukaryotes different from the origin of other lineages? That's another fantastic question. If you think about Darwin, it's descent with modification, right? And a lot of that is this vertical gene transfer. What that means is genes are passed down from parent to offspring. And what happens is you accumulate slow change over time, and over time, these lineages slowly evolve and you finally get new species. But what's different about the origins of eukaryotic cells is this. Two prokaryotes, a bacteria and archaean, they merged. And then from that merger, there were two prokaryotes living together. And then as they were living together, they evolved into a eukaryotic cell. And that's important because you didn't have this prokaryotic cell slowly getting larger and more complex and getting an early nucleus and some cellular membranes inside of it, early organelles. No, that never happened. You didn't have this slow change of a prokaryotic cell slowly changing into a eukaryotic cell. What you had is this merger of two prokaryotic cells, and then from that merger, that evolved into a eukaryotic cell or into eukaryotic cells, and that's quite different. Okay. Can you explain the importance of the proton gradient electron transport chain in ATP synthase? All right. That was back in cell biology, which is 201. And, we often need to repeat these things over and over. So I'm glad you asked that. So let's think about this. Every living organism needs energy. The energy currency of life is ATP. And there's several different ways you can make ATP. One is with an enzyme phosphorylation or substrate level phosphorylation. And the other way is through chemiosmosis. That's using a proton gradient and ATP synthase. And the idea is the protons flow through the ATP synthase, and as a result, that makes ATP. Now, you have to generate a proton gradient. That's where the electron transport chain comes in. And the electron transport chain uses the energy of the electrons to actively pump protons across a membrane, creating an electrochemical gradient. So if I were to draw it really quick, you got a membrane. If you've got a membrane, you've got complex in our mitochondria, you got complex 1, 2, 3, and 4, and then complex 1, 3, and 4. What they do is they start to pump these protons across that membrane. So now I have an electrochemical gradient. I've got more protons on this side of the membrane than this side. So that's a chemical gradient, but these protons are charged, and because they're charged, I get a positive charge right here on this side of the membrane versus a negative charge, and that's my chemical gradient, hence an electrochemical gradient. This system is out of equilibrium, and it wants to be in equilibrium. So you can harness this potential energy and allow these protons to flow through the ATP synthase. And of course, you can take ADP, which is like, think of it as a spent battery, and add a phosphate group to it and make ATP. Now, interestingly, every single organism on this planet has some form of this electron transport chain, some form of it. This is the one we find in mitochondria for eukaryotic cells that can do cellular respiration. And our source of high energy electron, of course, comes from the breakdown of organic matter like carbohydrates, like glucose, and lipids, like proteins. Not lipids, like proteins, sorry, like lipids. Okay. If you're doing photosynthesis, you don't have all these different complexes, but you still have the electron transport chain here, and your electrons come from water and your energy comes from sunlight. Like I said, eukaryotes, our energy is coming from the chemical bonds that we break down. So that's the importance of that there. And that goes back to this electron transport chain and chemiosmosis. The first cells on the planet almost certainly made ATP this way. So that's why we look for situations where you have a natural proton gradient where life is out of equilibrium in this environment, and you could concentrate organic molecules, including ATP. And that's why, going back to the very first question, why we think metabolism came before self-replicating molecules. Okay. When did life first appear on the Earth? There is compelling evidence that life started during the Hadean and may have popped up multiple times, but may have been wiped out by a giant meteor impact. Isaac, man, you're absolutely right. We don't have a lot of information about the origins of life on the planet and when life exactly got started. Several competing theories are out there. The one, we know that by 3.8 billion years ago, which is back in the Hadean, there is incredibly good evidence that life was getting started by then, because we already had these things called stromatolites. Now, I think there's some people that question the age of those stromatolites, but it's about 3.8 billion years. However, there's some compelling isotopic data that they find these ratios of light to heavy isotopes, and they think that only life could have created those ratios. Therefore, life got started much earlier, 4 billion years ago or so. And there are some chemists out there that are like, if life is going to get started, it's going to do it quickly. And life probably popped up multiple times and was repeatedly wiped out by giant meteor impacts and things like that. But what we do know is that whether or not life got started 4.1 billion years ago or 3.8 billion years ago, or even earlier than that, we know that all life on the planet today almost certainly came from a single population of the first living cells. And the reason why is because we have so many similarities, including using the same 20 amino acids, the same genetic code. Every protein is made in a ribosome, and we all use left-handed amino acids, right-handed carbohydrates. And that, because of these similarities, we assume that we came from one, basically, population of the first cells. When that first cells was, good point, we don't know. And yes, life may have definitely gotten started before that and been wiped out. We just don't have any evidence of it, so we don't really know. But that's a good question. People are trying to figure that stuff out, actually. Okay, Can you explain the contributions and limitations of the three theories of abiogenesis? Okay, I started out this review with those first three theories. Now think about this. The three theories are, we've got the prebiotic soup model. That's our early 1950s with Miller-Urey. We've got the RNA world hypothesis from the 1980s, and we have the metabolism first theory that came about, I'm going to say late 90s, but there's some people thinking it got there earlier. But here we go. The Miller-Urey experiment showed that the building blocks of life are easily made. It does not take much to create things like amino acids, pyruvate, acetaldehyde. These organic molecules are easily made from inorganic molecules. So there you go. That's the benefit of the Miller-Urey experiment. The drawback of the Miller-Urey experiment is it doesn't show you how you get those organic molecules and get something living out of them. Like I said, that goes back to how we think about life. The RNA world hypothesis was incredibly popular for a long time. And this goes back to the versatility of RNA molecules. RNA, unlike DNA, is a very versatile molecule. DNA stores information. It's very good at it. It's very elegant and simple. But RNA, being a single-stranded molecule, can not only store genetic information, RNA can also catalyze its own reactions and serve a variety of functions because it can assume multiple three-dimensional or tertiary structures. So in the 1980s, when they discovered that RNA can carry this genetic code and facilitate chemical reactions, including making more RNA, people got like really excited. They're like, wow, this is great, because life and biologists are focused on reproduction and evolution, because if you're getting reproduction, you can get evolution. The limitation, once again, great, I got a self-replicating molecule. I got viruses that will hit my computer if I'm not careful, self-replicate, and go out to every one of you. And I've been hit by viruses all the time. That doesn't mean that it's alive, right? Once again, the problem is, how do you get a system out of equilibrium with this environment? And the RNA world hypothesis does not explain that. So that's the metabolism first hypothesis, because that explains several things. It explains how you get a system naturally out of equilibrium with this environment. It explains why all life uses proton gradients to create ATP through chemiosmosis. And it explains like how you could get all of these organic molecules and concentrate them in an area that's very small for cells. What we still don't know is how you went from this metabolically active rock to a self-replicating cell. We still are like trying to get there. So got some stuff to still learn, but that's our best theory so far. Okay, explain how the Earth's atmosphere has changed since the origins of life. What caused this change? Well, we have some idea of what the Earth's early atmosphere looks like. We don't exactly know, but it's almost certainly much more carbon dioxide. There was probably some methane in there, a lot of nitrogen. And one thing that we do know is that oxygen levels were really low, like 0.01% of our atmosphere. And that is it. And that is very, very low. With the evolution of oxygenic photosynthesis several billion years ago, I think around 3.5 billion years ago, life began to pump out oxygen into the atmosphere. And oxygen is very reactive. It would have reacted with all the methane and ammonia in the atmosphere, cleaned it all out. And carbon dioxide over time became a lot less common. Oxygen went up. And today we now have an atmosphere that's basically 22% oxygen, 70-something% nitrogen, and everything else is in very small amounts. Okay, other changes that have caused other changes in the Earth's atmosphere. In the last 500 million years, we've fluctuated in amounts of oxygen and carbon dioxide, and there are several functions that are affecting the amount of carbon dioxide and oxygen in the atmosphere. About 400 million years ago, we started to get the first forest on the planet, and these first forests were photosynthetic, so they started pumping out gobs of oxygen. So by 300 million years ago, we had 30% oxygen in our atmosphere compared to about 22% today. So higher rates of oxygen in the atmosphere are always caused by photosynthesis, but very high amounts of oxygen were definitely caused by high rates of photosynthesis. And then about 252 million years ago, which was the end of the Permian, this is during the End-Permian Extinction, what happened was we had massive volcanism, and that spewed out lots of carbon dioxide into the atmosphere, and that over time warmed the planet a lot, and it disrupted our global ecosystems. And what happened is a lot of these forests collapsed, and a lot of the marine production collapsed, and as a result, oxygen levels plummeted from 30% oxygen down to about 12% oxygen. And then as those volcanism slowed down and the Earth's ecosystems recovered, we've stabilized, with about 22% oxygen in the atmosphere. And carbon dioxide levels tend to fluctuate between about 285 parts per million. You go back 300 or 400 million years, about 2,000 parts per million, but it's stayed within that range because of a complex play of geological processes and biological processes. Okay. Oh, I think I lost my place here. I moved. Can you explain how life can... Oh, man, I lost where we were here. Got a lot of your questions here. Can you explain how oxygen in the atmosphere is poisonous to many forms of life? How did early prokaryotes adapt to higher levels of oxygen? Well, think about it this way. Oxygen is oxidative. Oxygen will oxidize any organic molecule, proteins, carbohydrates, lipids, nucleic acids. So oxygen can damage your proteins, it can damage your cell membranes, and it can damage your DNA. That's why too high levels of oxygen is poisonous to life. I mean, it damages life. So one of the ways that life responded to high levels of oxidation, they created antioxidants, but they also evolved cellular respiration, using oxygen as a final electron acceptor. And what happens is that complex 4, we'll go back to this again. Here's complex 4. We got complex 1, little complex 2, complex 3, complex 4, which is the cytochrome oxidase. And what this complex 4 does is take the electrons, put them on oxygen, and then you get these protons coming through the ATP synthase, and you make water out of it. And how brilliant is that? Not only do you get more ATP made out of each organic molecule, but you also take oxygen, react it with hydrogen, and make water out of it, which is not toxic at all to life. So there you have it. One of the ways that life responded to higher levels of oxygen was to evolve cellular respiration. Okay. Yeah, so I've already said I'm going to draw a parsimony tree. I'll do that for you later. Okay. What were the adaptations of plants as the world became cooler and drier 300 million years ago? Yeah, so the big thing here is seed plants evolved. These are the gymnosperms. These include things like conifers, like pine trees and spruce trees and fir trees and cedar trees. Now, of course, these are modern day gymnosperms. So the early first gymnosperms were different than the modern ones today, but they evolved seed plants. And seed plants have pollen. And pollen is, you've seen pollen, it's that yellow stuff that gets all over your car in the springtime. And pollen has the male gamete inside of it. And what it does is it actually is an adaptation to drier environments, because pollen is very resistant to drought, and it no longer has to swim to the egg to get there. It can just grow to it, actually. And then also the advent of seeds, you put a hard waxy coating over your embryo, and this is also another adaptation for drier environments. And there were some other adaptations to the vascular system of gymnosperms that we'll learn in 304. Can you explain the theories of why animal life began to diversify after the Earth was already 4.1 billion years old? So if you think about it this way, that was 550 million years ago. That was the start of the Cambrian, specifically the start of the Cambrian Explosion. And what's going on here is that the Cambrian Explosion was a rapid diversification of life, especially animal life. And it happened 542 million years ago. And by the time the Earth was already 4.1 billion years old. That's like, the Earth is old when our ancestors finally diversified. There are lots of theories to explain this, and some of these work together. The first one, animals are eukaryotic cells. In fact, all multicellular life is made up of eukaryotic cells. And as a result, you need cellular respiration to have more complex eukaryotic cells. you need oxygen for multicellular life, and you need oxygen to energize life so that animals can have a lot more energy available to them to be more active. So higher levels of oxygen in the atmosphere. Second, um, animals have these unique set of regulatory genes called Hox genes. And Hox genes are very important because they lay out our body plan. And the whole point here is that, minor changes in your Hox genes, what they do is it can lead to large morphological changes. Okay. So with the advent of Hox genes, you can have rapid macroevolution by only having small changes in your Hox genes. Okay, another hypothesis is a rise of the predators. And this was an evolutionary arms race. So think about this. You're an early animal and you're just kind of worm-like. And all of a sudden you evolve eyes to see your environment and escape a predator. So your predator evolves eyes to chase down your prey. Your prey evolves a hard shell to avoid being eaten. Your predator evolves teeth. And what happens is you get this evolutionary arms race where life diversifies to either avoid the predators or to become a better predator and exploit different types of predation, to exploit different niches. And a lot of this was possible because of Hox genes that, like I said, you don't have to reinvent the wheel every time you need to make a small change in your body plan. You can have these minor changes in your Hox genes. So typically we think of the rise of animal life was this perfect storm of like higher oxygen levels, the advent of Hox genes, and the rise of predators and this evolutionary arms race. Okay. What is the most likely ancestor to all animals? It's called a choanoflagellate. Let me spell that out for you guys. Choanoflagellate. Choanoflagellate. So there it is, choanoflagellate. And these are protists. And a protist is a eukaryotic cell, single-celled. And I am not much of a drawer here, but this is the cell. It has this collar around it, and then this is a flagella coming out of the back of it here. And these cells actually look exactly like certain cells in sponges. And if you prod those cells that like you think they're going to get predated, they'll actually form a loose colony. So we think that predation may have actually caused the first single-celled organisms to form loose colonies so they couldn't be eaten. And if I said that, I've also got to be honest that some people think that multicellular life evolved to regulate oxygen levels to our tissues. Okay. Name the adaptations in plants that allowed them to evolve from small bryophytes to the first forest in the Devonian period. Okay, think about this. The first plants were mosses, bryophytes. These were plants that did not have leaves, they did not have roots, they did not have a vascular system, and they were soft. They didn't have lignified cell walls. So for plants to grow from these small mosses like this big into the forest we see today, you had to have several evolutionary innovations. One, you had to have a vascular system. And with that vascular system, what that does, you have xylem and phloem. The phloem connects the leaves to the roots and moves out all the carbohydrates and things the plant makes at every other cell. And the xylem carries water and minerals from the roots to the rest of the plant. So basically, the vascular tissue allowed plants to move materials large distances. Second, you had to have roots to anchor the plant to the ground. You had to have leaves to increase surface area for photosynthesis. And on top of your leaves, you have also a waxy cuticle, a waxy covering, which prevented desiccation, water loss. And you have these small openings on the bottom of the leaf called a stomata that you can further regulate how much water is moving through your leaves. And then lastly, but not least, the vascular system, but also lignin. And let me spell that for you so you know how to spell it. It's Lignan. And this is it, lignin right here. Lignin is it's like a proteinaceous type of complex. And lignin is a molecular complex that makes cell walls very rigid. Think about this, a 2x4 versus a spinach leaf. You can eat a spinach leaf all day long, and its cell walls, all of them are made up of cellulose. But you're not going to get a splinter from eating spinach. Don't eat a 2x4. And the difference between a 2x4 and spinach leaf is a lignified cell walls, and that allowed the plants to grow tall along with the vascular system. So all of these evolutionary innovations together in plants allowed for the first forest to evolve by the mid-Devonian around 400 million years ago. And that was pretty big because now all of a sudden the Earth, for the first time in 4.2 billion years, was greening up. Can you describe the adaptations of tetrapods to a cooler, drier environment about 320 million years ago? Yeah, sure. So if you think about this, at the beginning of the Carboniferous, like 375 million years ago, the Earth was a wet, swampy place. I mean, it was like Dagobah from The Empire Strikes Back, where you find ancient Jedi masters. But what happened was all of that carbon dioxide was slowly taken out of the atmosphere that was making it warm and wet, and it was sequestered. And it was lithified. Litho means rock, and it was turned into coal and oil. And as a result of CO2 levels in the atmosphere slowly being drawn down over tens of millions of years, well, the Earth's atmosphere cooled off, became cooler and drier. So animals had to respond to this cooler, drier environment. And the major evolutionary innovation at the time was the amniotes. And amniotes include both the Sauropsida and the Synapsida. Synapsida, of course, are modern-day mammals, and the Sauropsida are modern-day reptiles, including turtles, lizards, snakes, and birds, and crocodiles. And it also included a whole bunch of extinct things from the Mesozoic. But we're amniotes, and these innovations we have are things like internal reproduction. Think about a frog. They're amphibians, recently evolved from fish, right? Well, actually 375 million years ago. They get to return to water to reproduce. There are lots of exceptions to that, but they have external reproduction and have to return to water. With the evolution of amniotes, you have internal reproduction. So you no longer have to return to water to reproduce. Also, the embryo produces what's called the amniotic sac. This is a sac that surrounds the embryo and is derived from the embryo itself and keeps it in water, in a watery environment. And like I said, all amniotes, at least the ancestors, had to lay eggs on land. Because the other thing about amniotes is we skip this larval stage where we have to be in water. We're surrounded in our amniotic sac instead, and that's big. That's why lizards and birds can't lay their eggs in water, because their babies would drown, because we don't have gills. We don't have that larval stage like you do in amphibians. So those are the big ones. And then also our ancestors about 320 million years ago had scales, keratinized, and think keratin. This is keratin right here on your fingernails, and these keratinized scales prevented water loss as well. So those are the big evolutionary innovations of the amniotes that occurred about 315 million years ago, and very shortly split into the two groups that we know today as mammals and reptiles and birds. Okay. What are the problems with the metabolic first theory? First. Okay. Biggest problem with that is, I've said this before, so I'm going to do it really quickly here. What we don't know is how our first cells acquired the ability to store all the genetic information in DNA, and how you replicate that DNA. And that's something we're still working on. We haven't figured out how life became replicating. So somehow it did. We're still working on it. We know that RNA was certainly involved with that, because as we know from the RNA world theory, so RNA is a self-replicating molecule that can store genetic information. So somehow there was a leap between this RNA to DNA, and then the cells are actually able to leave their birthplaces in these metabolically active rocks. Okay. Can you explain the importance of forest for diversity? Yeah, it creates habitats. The more habitat you have, the more niches you have. So as you got vertical structure, you had more places for things to live, and having roots increased soils. So the first forest greatly diversified life on this planet, because as the forest expanded and trees evolved, they stabilized the soil, so that allowed for more trees to grow, and just created lots and lots and lots of habitats for lots of animals to evolve and exhibit and live on the terrestrial surface. Okay, mass extinctions. The root cause of mass extinctions almost always is climate change. Whether or not that's brought on catastrophically by a large meteor impact, like we saw at the end of the dinosaurs, or from periods of intense volcanism, like we saw in the Deccan traps and in the Siberian traps, or from the movement of continents that slowly slide across the globe, but ultimately, or from the way animals and plants change the atmosphere, especially plants. So these things can all factor into how climate changes. And we know that every time the climate changes rapidly, things go extinct fast. So climate change is the big one, and what the causes of those climate change can be quite varied. From, like I said, the slow movement of the continents to animal life on the planet, to intense volcanism, to meteor impacts. Okay, which phylum should we know characteristics of? All the ones I went over in class. There you go. That would be the cnidarians, the arthropods, the mollusks, the chordates, and the echinoderms. And not only that, you want to make sure that you know the 4 subphyla of arthropods, which are the hexapods, which include insects, crustacea, myriapods, and the chelicerates. And you want to know something about the vertebrates as well, especially like the jawed fish, the cartilaginous fish, the ray-finned fish, bony fish, yeah, tetrapods, amniotes. You want to make sure, especially amniotes, and you want to know mammals. Okay, can you describe adaptations? I already went over that one. What most likely caused the End-Permian Extinction? Yeah, so there's some cool stuff about this one. So 252 million years ago was the greatest mass extinction the Earth has ever seen. And we think it actually happened in 2 waves. So if you want to get the A answer on this is how you answer it. The Siberian Traps started off with this intense period of volcanism, which spewed tons of ash and sulfates into the atmosphere, which plummeted the Earth's temperature. And that caused a first round of extinction event. But that was followed by rapid warming. So after like a period of a couple decades of really cool temperatures from all the soot and ash and sulfates in the atmosphere, reflecting sunlight back out into space, like I said, that cooled the atmosphere. What happened next was, well, those volcanoes, they didn't stop going off for tens of thousands. I think they almost lasted 100,000 years. They just pumped gobs of CO2 into the atmosphere, and that spiked temperatures rapidly over the next 100,000 years. So initial wave from rapid cooling, and then another sustained wave from the warming that took place. So by the time that the Triassic was getting started, after the Siberian Traps ended, the Earth was hot and dry and with low O2. And like I said, that really did a 1-2 punch on all life on this planet. I mean, like 90% of all marine life or more went extinct, and like 70% of all terrestrial life went extinct. Okay, can you explain how life changed the atmosphere in the Carboniferous? How'd that affect the climate? Carboniferous, so where we get all our coal and oil from, right? So there were the first forest on the planet. It was a wet, swampy world. Like I said, it's like Dagobah. Think of like Master Yoda. And what happened is, as all this plant was growing, it would fall into these warm, watered areas, and you need oxygen to break down organic material back to carbon dioxide and water. Without a lot of oxygen in the water, in the swampy areas, the organic material became lithified as coal and oil. So that carbon dioxide was slowly removed out of the atmosphere by the actions of plants, by photosynthesis. Over millions of years, enough CO2 was removed that the plant went into an ice age, much like today, as carbon dioxide went from 2,000 parts per million down to about 285 parts per million. Okay. Over the problems with each theory of abiotic, I've already done that. Why are tetrapods in the same lineage as lobed fin fish? That's a good question. Basically, we're modified fish. I know, it's pretty cool. We're modified fish. But there are these things called osteichthyes, the bony fish, and there are 2 major lineages, the ray-finned fish and the lobed fin fish. There's like all of 9 species of lobed fin fish in the world today. But we evolved from a lobed fin fish about 375 million years ago. And we know this based on the fact that a lobed fin fish actually has a shoulder girdle here with a scapula, and they have in their fin, this is a modified fish fin, believe it or not, this is a modified fish fin. They've got a pattern of 1 bone, 2 bone, lots of bones. And they have paired appendages, so this would be the pectoral region. And then your legs represent the pelvic fin, or the pelvic region, which also has a pelvic girdle and a series of bones. They go one bone, two bone, lots of bones. And sorry, let me take my phone out there. And because these lobed fin fish have that, we assume that we came from them as well. Also, we have similarities in the fact that our lungs come off of our esophagus, but specifically back here, there's a trachea. You can see it right there. This goes into my lungs. And there's an epiglottis that covers it every time I swallow. But basically, my lungs are derived off of my esophagus. These lungfish also have lungs, lobed-fin fish or lungfish, that come off of their esophagus as well. And there's all kinds of other supporting evidence based on the fins, the placement of the lungs, and our DNA also supports that connection as well. Okay. So I've already answered the theories of why animal alive. What were the first land plants and when did they evolve? You know, the first land plants were those small little bryophytes that first evolved, we think, in the beginning of the Silurian, like almost 440 million years ago, 430. We're still trying to place a good date on when those plants first started to colonize the land. And it took them a while before they evolved into the vascular plants. But the first plants were the bryophytes. And that was, you know, back in the Silurian. Okay, I already did the change in the atmosphere, implicated in several mass extinctions. We've done that. It's climate change. Please describe the adaptations of, I think I've done all these. Why were dinosaurs dominant over our mammal ancestors? That's a really good question. Because, you know, if you were to go back in time to 255 million years ago, at the end of the Permian, is the end of the Paleozoic era, which is something you want to know, the dominant land animals were these synapsids. They wouldn't have looked like mammals. You'd have mistaken them for a reptile, but the differences in their skull would have been very revealing that these were not reptiles known as Sauropsida. They were clearly the ancestors to mammals, and they were dominant. And then it comes the End-Permian, the Great Dying, right? And these things all get wiped out. And then all of a sudden, in the Triassic, you still have synapsids, and you've got a lineage of reptiles called the archosaurs, which include crocodiles, modern-day birds, and dinosaurs, and pterosaurs as well. But in the Triassic was very different than the end of the Permian. At the end of the Permian, you know, it was you had high levels of oxygen, like 30% or more, or not more, but around 30%. And synapsids, we have a two-way lung. We breathe in, and then my air goes into my lungs, and then it goes back out. So I'm mixing fresh air with old air. Now, the archosaurs, we think, now not everybody agrees with this, but there's good evidence that suggests that the archosaurs had what we call a one-way lung. And in the Triassic, oxygen levels were much lower. They were like 12% of the atmosphere compared to 22% today or 30% in the Permian. And having a one-way lung, the air moves in your lungs in only one direction. So you never mix your fresh air with your old air. And because of the one-way lung, they were much more efficient at extracting oxygen from the atmosphere. So that was very helpful, and especially when you only have 12% oxygen in your atmosphere. That's like us standing at 15,000 feet today. So we don't do very well that high. Another thing that the dinosaurs had is their legs. They were bipedal. They ran on two legs. And what that did is that separated the breathing from their movement. So if you've got sprawling legs out here, you're moving like this, you're changing the shape of your lung. So your movement is tied to, you know, moving air in and out of your lungs. But once we got our legs up underneath us, and including the archosaurs, that made breathing much more efficient. Now, synapsids probably had, the ancestor almost certainly had a four-chambered heart, even back in the Triassic, but the archosaurs almost certainly had a four-chambered heart too. And that didn't separate them out as being better than the synapsids, but the fact that the archosaurs, these dinosaur ancestors, had this four-chambered heart allowed them to have higher blood pressure, which allowed them to be more active by being larger, and it also prevented the mixing of oxygenated and deoxygenated blood as well. So you take being bipedal, which separates movement from breathing, a one-way lung and a four-chambered heart, and that made them, we think, definitely more competitive than the ancestors to mammals. Even though mammals had a four-chambered heart, we were really limited by our two-way lung at that time. And as a result, dinosaurs got the jump start in the Triassic and became the dominant species for the next 170 million years. Okay. All right, I've already gone over this. Implication of several mass extinction, why life is extension of geological processes. I've done that. I just went that one. Oh, man. Okay, so it jumped all the way ahead. Also, are cnidarians, mollusks, and arthropods a sufficient answer for info? I don't have the questions in front of me. You got to ask a real question here. I don't have the question. So you got to ask me like something about the material. I can't just answer that question off the top of my head. How are plants able to evolve from small bryophytes to the first forest in the Devonian period? Remember, I answered that one earlier, and that has to do with the vascular tissue evolving roots, leaves, cuticle, stomata, and lignified cell wall. So I talked about that earlier. Explain why tetrapods are in the same lineage as lobe-finned fish. We evolved from lobe-finned fish, so we're in the same lineage. When you talked about when life began, you mentioned multiple times that it might have been before the Archean. Do you want us to answer with that or something else? That's a really good question. I mean, basically, if you tell me, life evolved 3.5 billion years ago, that's a conservative estimate. If you say 3.8 billion years ago, that's probably the most accurate estimate we have. But if you mention life, we know for certain life was around 3.5 or 3.8 billion years ago, and almost certainly evolved as early as 4.1 billion years ago, based on some isotopic signatures and those ancient rocks. I'd go for that. You could answer that. Okay, explain how the origins of eukaryotes is different from the origin of other lineages. Got that. Explain why tetrapods are in the same lineage. Got that one. When you talk about when life began, you mentioned multiple times. Okay. What is the difference between therians and the first amniotic animals? Does only one involve lateral gene transfer via bacteria? Okay, the difference between therians and the first amniotic. Okay, well, I think I can answer that one. Okay, the first amniotes, clearly we all had internal reproduction and we produced this amniotic sac. But the first amniotes laid eggs. Mammals, of course, don't do that. We give live birth. And one of the most mammalian traits we have, besides producing milk, is we have a placenta that connects the mom to the growing embryo. And interestingly, when we did the whole genome of mammals a few decades ago, one of the things we discovered was that the genes involved with developing the placenta, and specifically the placenta embedding into the endometrium, which is the inside of your uterus, those genes were not mammalian in origin. They actually came from a retrovirus, which meant that we got genes to help grow a placenta from a retrovirus, which is an example of lateral gene transfer, meaning we got those genes from something in our environment, in this case, a retrovirus, versus being passed down from parent to offspring. Okay. Can you please define an outgroup, node, fork, branch, and a clade? Oh, come on. All right. I hate to turn down answer questions, but basically, this is just looking at a phylogeny. I'll draw one very quickly. But on a question like that, one of the things that you can do is just Google those definitions, and by Googling those definitions, it's easy to see what they are. So I drew a quick phylogenetic tree right here. This dot right there, that represents a common ancestor, okay? That's a common ancestor. That's a node. That's a branch point. We could also draw the exact same tree to look like this as well, okay? So this point right here represents this point right here. Now, our two closest related groups would be, this would be sister taxa, and this would be an outgroup. And I So there's your sister taxa because they're the most closely related. They share the most recent common ancestor, and this is the outgroup here. And we could draw it here like sister taxa, and this would be the outgroup here. And you could, like I said, you can draw the trees the same way. Okay, so a lot of times if you have problems with just definitions, one of the easiest things you can do is just, like I said, just Google the definition and go to Wikipedia. Wikipedia is awesome for answering that. Okay. What is Tor name? I don't know what that is. What is your name? What state are you from? Yeah, okay. I'm Tom Kennedy Science. I live in New Mexico. There you go. What is the most likely ancestor to all animals? I did that earlier. That's a choanoflagellate. Did amniotes split into diapsida and synapsida or sauropsida and synapsida? Good question for a little clarification here. Basically, diapsida means two temporal fenestra, right? And sauropsida, well, diapsids are sauropsida. So you could have said, you know, amniote split into synapsida and sauropsida, and you can replace sauropsida with diapsida for most cases. So you could have said they split into synapsida and diapsida. Dimetrodon. Dimetrodon is a pelycosaur from 310 million years ago. Dimetrodon was a synapsid, and it was on the Earth millions of years before dinosaurs ever evolved. And it's an ancestor to mammals, not to sauropsids. And that's why it's not a dinosaur. And for questions like that, I mean, what I would do is just go to the Wikipedia and look up Dimetrodon, and it'll give you all the information on it. Okay. Why it is highly unlikely that one prokaryote or an early proto-eukaryote did not phagocytize another prokaryote in the evolution of eukaryotes? Hey, now that's a good question. I like these questions where they're much more conceptual versus just given a definition of something. Like I said, I would strongly encourage you guys to look definitions up on the Wikipedia. But here it is. You know, why didn't we see prokaryotes, you know, doing this number right here? Okay, why didn't we see that? Well, and why didn't we see, you know, this last one phagocytizing another cell? And that's my phagocytizing right there. You're taking the membrane and moving it around another bacteria and then incorporating it. Let's see if we can do that up here. You know, how come we didn't see this? Oh yeah. And then there's our prokaryotic cell getting larger, more complex, nucleus phagocytizing the mitochondria, and then here's your cell nucleus, here's your mitochondria. How come we didn't see that? First of all, eukaryotes are almost 1,000 times larger than prokaryotes. Now, contrary to what you learned in cell biology textbooks, I'm almost always wrong on this. And the reason why is surface to volume ratio does limit the size of a cell, of a prokaryotic cell, but not because of diffusion. Cells become limited by energy production because all cells make ATP across their membranes. So as a prokaryotic cell gets larger and larger and larger, the volume increases at the power of 3, length, width, and height. And the surface area gets larger by the volume, not by volume, but by power of 2, length and width. So as a cell gets larger and larger and larger and larger and larger and larger, right, what happens is the volume gets much larger than the surface area. So when a cell starts getting larger like this, they run out of energy. They can't make enough ATP. Okay, so that's why you don't have a proto-eukaryotic cell. The other reason why we don't see a proto-eukaryotic cell engulfing anaerobic bacteria is because to engulf something is called phagocytosis. You need actin filaments and microtubules to move your membrane around another cell to engulf it. No prokaryote has actin filaments and microtubules, and we see no evidence of prokaryotes doing something like phagocytosis. We only see that in eukaryotes. And we have no reason to believe that 2 billion years ago, prokaryotes would have had this amazing ability to do phagocytosis, and when they don't do it today. and they would just run out of energy. And we don't see them with a cytoskeleton capable of doing that. So we have no reason to believe they could do that 2 billion years ago. That would be like going back to the beginnings of the agricultural revolution 10,000 years ago and expecting to find a Boeing 747. That just doesn't happen, right? So that's the main reason why. So what we do know is that bacteria do bore into other prokaryotic cells and can reside in there. And that's almost certainly what happened. Okay. Were humans always set up to cause the sixth mass extinction? Man, probably. I think we've been causing extinction events since the Pleistocene. People came over to the U.S., over to North America, about 15, 20,000 years ago, and we almost caused the collapse of the large animals on this continent. So, man, we've been causing extinctions since day one. So, yeah, once where our technology got bigger, got better, and we were just doomed to do it. Okay, phagocytosis did not occur because it requires more energy than can be created without mitochondria, prokaryote, cytoskelet complex enough. Oh, cool. Yeah, thanks, Ashton. Okay. Oh, come on. Every time I move down, it goes all over the place. They need to work on that. Okay. Is the only difference between the synapses and diapsids in their holes? I don't know what that means. I can tell you that there are several differences between them, and one of them is in the temporal fenestra. Fenestra means windows, and those are openings in the skull, whereas the first synapsids had one of those openings, fenestra, and diapsids or the sauropsida had two of them. There's also differences in the way our jaws are, the way our teeth are set, and our inner ear as well. So it's not just the differences in the holes. And let's use a little bit more scientific knowledge, scientific jargon than that. Okay. Well, we need to know the dates for certain eras and epochs. Well, I told you you need to know the dates for the eons and the eras. The eras of the Phanerozoic eon, and you also want to know the Cambrian Explosion, and it'd probably be helpful to know like Jurassic and Cretaceous too. Can you list a major that's a lot, haven't we? I mean, I went over that like a lot of times in class. So let's talk about them. Okay. When it comes to life, I mean, life has been steadily getting more complex. So how did we go from like metabolically active rocks 4 billion years ago to the wide diversity of life we see today? And there were lots of evolutionary innovations along the way that occurred. Things like the ability to replicate DNA, oxygenic photosynthesis, pumping oxygen to the atmosphere, the evolution of aerobic respiration, endosymbiosis, and the evolution of eukaryotic cells, the evolution of multicellularity in animals. Then as we get into more modern times, other major evolutionary innovations include things like vascular plants, flowering plants, which is also a major adaptation of plants that allow for incredible diversity through adaptive radiations. Other major evolutionary innovations, things like tetrapods, jaws, lungs, the amniotic sac, internal reproduction. So these things are some of the major evolutionary innovations. Oh yeah, and endothermy was a big one too. So I hope that helps out. Okay. Describe the factors that cause a dinosaur's extinction. End-Permian extinction cause reset of competitive exclusion, which is what was keeping mammals from moving into dinosaur territory. Yeah, so the dinosaurs went extinct, and it's probably a 1-2 punch. It was the Deccan traps that went off that started changing the climate, acidifying the oceans again, and then a large meteor, the Alvarez impact, caused that. So that definitely probably caused the extinction event. Okay. I already talked about why life is an extension of geological processes. Can you explain the importance of photosynthesis as a major evolutionary innovation? Yeah, that's a good question. And the reason why, there's several things. Think about this. All the oxygen in our atmosphere is a result of photosynthesis. That helps stabilize the planet in terms of like creating the ozone layer, protecting our water, and the extra oxygen energized life, and it paved the way for things like aerobic respiration, eukaryotic cells, multicellular life, and specifically animal life. Also, photosynthesis is very important because it makes energy and nutrients available to us. So every carbon atom in your body was fixed by a plant doing photosynthesis. So that made that carbon available to you. And also, all the energy you have in your body came from the sun, but was transformed from the kinetic energy of sunlight to the chemical energy found in the bonds of the foods that you eat. So not only does all the carbon in your body was fixed by a plant, also all of your energy was made available to you by photosynthesis as well. So those are the major reasons why photosynthesis was a major evolutionary innovation, because our world would be very, very different today without photosynthesis. Yeah, uniformitarianism, as it's related to uniformitarianism. Uniformitarianism is this idea that, it's not an idea, it's this theory that basically the principles of science, the laws and the theories of the way the universe works operate the same everywhere in the universe, and they operate the same as they do today as they did in the past or into the future. So, flowering plants. That was a major evolutionary innovation for diversity. To give you an example, there are 300,000 species of flowering plants today and 640 species of gymnosperms. The reason why is that flowers attract pollinators. So as flowers diversified, so did the pollinators. You got pollinators coming to flowers. You've got predators on those pollinators. You've got parasitoids. You've got animals that evolve to go after different types of fruits. So it's almost as if diversity gives rise to more diversity. Okay. I already talked about why dinosaurs became dominant. Can you explain the origin of eukaryotes? I already did that. Is starfish an echinoderm or cnidarian? Starfish are echinoderms, and they have that kind of a spiny skin. And the big difference is that they both have radial symmetry. But cnidarians, that radial symmetry is an ancestral trait, whereas in echinoderms, like a starfish, it's a derived trait. And cnidarians also only have 2 germ layers, endoderm and ectoderm, whereas a starfish actually has 3 germ layers, endoderm, mesoderm, and an ectoderm. Okay. All right. And then this question here is probably my last one. Explain the importance of the Cambrian explosion. Yeah, I mean, all modern phyla today, the ones we talked about, cnidarians and echinoderms and chordates and mollusks, all arrived very quickly within this short time period called the Cambrian explosion. Okay, well, thanks for all your questions. Some of those are really good. Like I said, in the future, learn to look stuff up on the Wikipedia for simple definitions, and then check yourself rather than just simply asking them. And then, and then these more conceptual questions like the importance of mass extinctions for evolution. When somebody slipping one in here at the last second here, those are better ones to ask here. And the importance of mass extinctions in evolution is that it wipes out all the dominant species, kind of cleans the slate, so to speak, and allows for new lineages to become dominant. And how can we date ancient rocks and fossils? And how could you explain radiometric dating? We use radiometric dating, which is every element has isotopes that are unstable and they're radioactive because they decay into daughter elements. And what you do is you take rock samples and you look at the amount of radioisotopes in them and compare that to the daughter isotopes. And then based on that comparison and knowing the half-life of those radioisotopes, you can calculate an age on that. And one thing on the test, remember this, look for radioisotope dating, not radiocarbon dating. Radiocarbon is one isotope is good for like 50,000 years, and that's it, versus radioisotope dating. We can put dates on things billions of years old. And Kayla and Jacob, you guys, you're absolutely welcome. I hope this is helpful. And good luck tomorrow on the test. And I will see you guys tomorrow. It was a great semester. I really enjoyed teaching you guys some evolution. And I hope that with all the whirlwind at the end of the semester on going through other diversity, that I just kind of like piqued your interest in all the different types of animals that are on this planet. And tomorrow when you have to talk about your favorite animal, try to give us something cool that we may not know about and go do some research on it. Okay. Thanks, Abby. Yeah, thank you. You guys are totally welcome. I enjoy doing this, and have fun, and I will see you guys tomorrow.